All living organisms depend upon features of their natural environment for survival in one way or another. When conditions change, organisms respond accordingly by increasing or declining within their current ranges, or expanding into newly suitable territories. Some organisms are endowed with special characteristics, which cause them to be highly sensitive to specific alterations in their microhabitats, while being entirely resistant to a number of others. By studying the past and present spatial distributions of these so-called indicator species, scientists have a means of identifying and examining minute or historical environmental changes which humans could not ordinarily perceive. In this series of videos, I will introduce you to a project designed to database and publish much of the valuable information accompanying the vast collections of lichens and bryophytes stored in the nation's top herbaria, for the purpose of expanding the usage of these two groups of organisms as indicators for studying global environmental change. Lichens and bryophytes are two distantly related groups of organisms, which, through convergent evolution, have come to share several unique and interesting physiological and ecological characteristics, which cause them to have an unusually intimate relationship with their natural environment. Unlike their larger cousins, macrofungi, and the vascular plants, lichens and bryophytes most often do not have large or particularly showy growth forms or reproductive structures, and do not typically root in deep, nutrient-rich soils. Instead, they tend to colonize the outer surface of hard, exposed, or relatively nutrient-poor substrates such as rocks and tree bark, with a relatively short and dense mat of living material. Since they lack proper roots and specialized vascular tissues and have no protective outer cuticle, moisture and nutrient uptake is largely atmospheric, and essential gases such as oxygen and CO2 disseminate directly into, out of, and between cells. This extreme exposure of lichens and bryophytes to the forces of nature causes them to be highly sensitive to many external stimuli, such as temperature, moisture, air quality, and nutrient availability. The second principal reason why lichens and bryophytes can serve as good indicators for studying environmental change has to do with their exceptional interspecies diversity. Although several general characteristics are shared between many lichens and bryophytes, a seemingly infinite amount of variation still exists in the minute structural details and physiological demands of each individual species. These distinctions, while interesting from purely botanical and evolutionary perspectives, also cause these organisms to respond independently to a variety of environmental cues, which greatly bolsters the range of environmental parameters which can be investigated using lichens and bryophytes as indicator species. For centuries, naturalists have been captivated by the small but ubiquitous life forms classified by scientists as lichens and bryophytes, as evidenced by historical collections dating back to as early as the 1700s. Since then, rigorous scientific investigation of these organisms has only intensified over time, resulting in the accumulation of considerable quantities of botanical specimens, which are stored in places known as herbaria. There, these collections are constantly scrutinized, renamed, and reassigned to taxonomic groups as modern scientists use new tools and techniques to revise our knowledge of the evolutionary relationships between organisms. These collections have always been an invaluable asset for systematists, who study evolutionary patterns by examining the physical and molecular structure of specimens. However, recent advances in geographic information sciences, species distribution modeling, and statistical inference regarding spatial relationships have allowed the specimens stored in herbaria to become an increasingly more useful resource for studying fundamental ecological principles and patterns of environmental change on a global scale. Taking advantage of the innate and highly species-specific sensitivities of lichens and bryophytes to changes in their immediate microhabitats, scientists have already been able to establish strong correlations between the distributions of individual lichen and bryophyte species and a range of critical environmental pressures, including acid rain, air and water pollution, eutrophication, climate warming, fire regime changes, and many others. This groundbreaking work has set the stage for the widespread use of lichen and bryophyte collections to monitor past and present environmental change. However, the predictive power of these types of investigations has been largely limited by the poor availability of historical collection information to the general scientific community. The TCN Lichen and Bryophyte Databasing Project will be a collaboration between 60 institutions in the US, Canada, and Mexico to digitally catalog an estimated 2.3 million North American lichen and bryophyte specimens and combine their reference files into two publicly accessible databases, the Consortium of North American Lichen Herbaria and the Consortium of North American Bryophyte Herbaria. For the sake of maximizing efficiency, the project will focus on databasing the species determination and collection information for each specimen, while utilizing the latest archival and imaging technologies and automated data entry techniques. Because of the established scientific applications of using these organisms as indicator species, we believe that databasing North American lichen and bryophyte collections as part of this collaborative multi-institution project will do more than simply improve the accessibility of historical specimen information. Some examples of the types of activities which we hope to foster through this project are documenting lichen and bryophyte distribution changes over time, creating maps showing land use, species distributions, and environmental change, using known indicator species to identify environmental issues and areas of conservation concern, documenting effects of past conservation and habitat restoration action, offering additional high-quality data for the exploration of novel correlations and identification of new indicator species, and finally, generating more detailed hypotheses regarding the past and present spatial distributions of environmental changes in North America. As with any large collaborative undertaking, the success of this databasing project will inevitably depend upon the continuous support of all contributing parties and participating institutions. Therefore, in the interest of supporting our fellow collaborators in this project, we have provided in our next video a detailed description of the workflow procedures used here at the New York Botanical Garden to process lichen and bryophyte specimens for digitization. We hope that you find both of these videos helpful for introducing your staff to the project and for guiding the design of your own efficient workflow procedures. Thanks for watching.